Hello students, welcome to Sorosar classes. Today our topic is biotechnology and we are going to discuss the conceptual development of the principles of genetic engineering that is also a part of biotechnology. Genetic engineering is also a part of biotechnology and we are going to discuss about the conceptual development of the principles of this genetic engineering. So there are several principles of this genetic engineering and we are going to discuss this step by step. The first one is the technique of genetic engineering includes the technique of genetic engineering includes the formation of recombinant DNA that is known as rDNA. So it includes formation of recombinant DNA that is rDNA, the use of gene cloning, and gene transfer. So the first use of this genetic engineering is the formation of recombinant DNA, the use of gene cloning and gene formation. Now the second one is it checks the limitation and permits to isolate only one or a set of desirable genes without introducing undesirable genes. So it checks the limitations and permits to isolate only one or a set of desirable genes or a set of desirable genes without introducing the undesirable genes into the target organism. So it basically permits the introduction of the undesirable genes into the target organism. Now the third one is the piece of DNA is not be able to multiply itself, to multiply itself in the organism, in the organism, and can multiply only when, and can multiply only when it gets integrated into the genetic material, only when it gets integrated into the genetic material, into the genetic material of the recipient, of the recipient organism. The next one is the alien or the foreign piece of DNA is added to the recipient chromosome, which possesses the ability to replicate, the ability to replicate. So, in the last stage, the piece of DNA, we said that the piece of DNA is not be able to multiply itself in the organism and can multiply only when it gets integrated into the genetic material of the recipient organism. And then after the third stage, the line of the foreign piece of DNA is added to the recipient chromosome, which possesses the ability to replicate. Now, the next principle of genetic engineering is it also called the cloning. It is also known as cloning, that is forming multiple identical copies. That means forming multiple identical copies of any template DNA. So cloning means the forming the multiple identical copies of any template DNA. Thus, genetic engineering is alternately called the recombinant DNA technology. Thus, genetic engineering can also be called as recombinant DNA technology, that is rDNA technology, or gene cloning. Now, the next point is, if the sequence is present, if this sequence is present, the DNA will replicate. The DNA will replicate. Okay, now let me add a new page. Will replicate if it is absent. If it is absent, the DNA will not replicate. The DNA will not replicate. So, in brief, the replication of DNA depends on this genetic engineering. Therefore, the aligned piece of DNA fails to replicate until it fails to replicate until it gets integrated with the chromosomal DNA. It gets integrated with the chromosomal DNA having the origin of replication. So, these are the main conceptual development of the principles of genetic engineering.